Good morning, everybody. So welcome everybody again to the second day of our workshop. I would like to welcome and to introduce Dr. Marcelo Bozza. So Dr. Marcelo Bozza is an old friend of our graduate program in immunology here. He was already in some previous editions of our workshop. And I'm gonna give you a brief, not so brief introduction about Dr. Marcelo Bozza. So Dr. Bozza is a physician. He got his medical degree from the State University of Rio de Janeiro. He also has a master's degree in parasite biology and a PhD in cellular and molecular biology by Fiocruz in Rio de Janeiro too. He also, after that, did his postdoctoral training at the Harvard Medical School at the USA. He is currently a full professor at the Federal University of Rio de Janeiro at the Department of Immunology from the Paulo de Góes Microbiology Institute, where he participated in the establishment of the graduate program in immunology and inflammation. Dr. Bozza is also a permanent member of the Brazilian Academy of Sciences. He also acts as a reviewer for several important journals such as JI plus Pathogens, PNAS, Blood, eLife, and among others in which he has already contributed to several important papers. And finally, Dr. Bozza is an active member of our Brazilian Society of Immunology, in which he was also part of the board of directors between the years of 2014 and 2015. And it's a great pleasure for us to have you here, Marcelo. He's gonna give his talk that he sent us a mix of French and English, right? Liaison Dangereuses, microbiome, tolerance, and gut inflammation. So please, Dr. Marcelo.
Thank you very much for your kind introduction. It is true that I'm very linked to the PhD program of USP Ribeirão Preto, where I have so many good friends, and I'm very good friend as well of the most important person of this PhD program, Ana, a secretária do programa. That's a key person for the success of your program. And I am also connected to previous students that now are colleagues. So I think it's a privilege to be here today. It would be better if it was in person, if I could be there in Ribeirão Preto, enjoying the meeting, the whole meeting, and talking to people and drinking a beer and discussing science and so on. So today I will present a story that you will see, it's kind of full of surprises. And it's nice when science brings you to a space where uncertainties are prevalent. So you kind of find yourself amazed by things that you did not predict. So I think that it's an interesting story, and as you will see, I think it's full of surprises. So I will share with you my screen. And please let me know if you can see these well. And hold on, let me just take these out. Okay. So Diego, can you see the?
Yes, it's fine, Marcelo.
Okay. So as you know, our world is a microbial world. I mean, it has always been, and it will be even more. And the thing is, we usually, or we have been thinking about infection mainly associated to pathogens. But of course, we know that this interaction of microorganism and a host, it can be of different ways. So it can be just no infection. So a microorganism might not be able to infect a specific host. It can be in commensal association, or some microorganisms can act as pathobionts or as pathogens. And they might be bacteria, fungi, virus, or protozoa. And of course, that one microorganism might also affect the way a different microorganism interacts with the host. So one infection might affect the host, and affect the interaction with a different microorganism. So it's a complex association, and of course, we know that this is dependent on a evolutionary process. And if we think the way hosts can deal with infection, at least in terms of defense strategies, we know that there are like two main different strategies, and they are conceptually different. So one is, so I think that someone has the audio open. If you could close the audio, would be nice. So one is resistance. So resistance basically is the way that host deals with infection by reducing the burden, the pathogen burden, or the infection burden. And basically the question is how we fight infection. And as you can see here, resistance will deal with the idea that one would be able to recover health by reducing the pathogen burden. And the main way that we do that is through the immune system. As you can see here, in different species, we have basically in vertebrates the innate immune system, and vertebrates will also have the adaptive immune system. So that's one way to deal with infection. The immune system is not the only one associated with resistance. There are many other different systems that contribute to resistance, but of course, the immune system is the main system associated with this function, that is, control the number of pathogens or microorganisms. But there is different ways, and this story is remarkable, is the story of Mary Mellon, that was a cooker in New York by the end of the 19th century. And it was found that she was responsible by several infections, cases of Salmonella typhimurium. So she was considered the first healthy carrier case in the United States. But the point I want to make here is that despite she was highly infected, she was not sick. Actually, she was, in very good health. And that was remarkable. So basically what I'm trying to say here is that in the case of, specifically Mary Mellon, she was not very good in terms of resistance, meaning she was not able to eliminate the pathogen, but still she was very healthy. So that indicates that there is a different strategy to deal with infection, that is disease tolerance. So disease tolerance, as you can see, so it's a way associated to reduce the negative impact of infection, but this is unrelated to the resistance mechanism. So it does not really directly affect a pathogen burden. And the question would be how we survive infection. And as you can see here in this figure, so to a certain pathogen burden in arbitrary unit here, you can see that, let's say in 10 to the 7, the person in green is still healthy, is going to work, to the university, and so on. So the one in orange, with the same pathogen burden, it's severely sick. And the one in red is, barely surviving, so it's almost dying. So this shows that, the ability to control the infection is not the only thing important when we deal with an infection organism. And as you can see here, again, this is also conserved through evolution, and is dependent on genetic factors. So as you can see here, Drosophilas that are a wild type or with a mutation, they have a different survival curves. But when we look the bacterial counts, you observe that they are similar. So they have the same bacterial counts, but the wild type die faster than the mutated one. So it is conserved through evolution, and it has a genetic determinants. But also, as you can see here, in this a model of infection with flock house virus in Drosophila that has a Wolbachia, or they are not colonized by Wolbachia. As you can see here in the top figure to the right, the ones that have Wolbachia, they survive more to the flock house virus infection compared to the one that is not colonized by Wolbachia. And again, when we look the pathogen load, the viral load, we observe that both groups have similar amounts of virus. So there are different mechanisms associated with disease tolerance, as you can see here. So there are many groups of systems associated with the stress response. And, upon infection, these stress response programs eventually leads to metabolic adaptation and tissue damage control and disease tolerance. So the individuals or the animals are fine responding to a specific infection. But these can also lead to maladaptive response. And then we go to damage response systems that are, in a different level compared to the stress response ones. And this can repair the damage and go back to the tissue damage control and disease tolerance, or this can eventually be insufficient to control the damage, and you observe cell death and tissue damage and what we call disease. So one could say that in each individual host, in one species, the coordination of these both abilities to deal with infection, resistance, and tolerance will define the community composition of commensal or symbiotic microorganisms, as well as infection agents that can be or tolerated, controlled, eliminated, or will eventually cause disease. So all these evolutionary aspects, suffer a major change when we think about our more recent evolution as humans. So if we go back to 12,000 years ago, basically we would live, in the savanna, in the forest, as small groups. And then, we suffer a major transition with the agricultural revolution. And of course that, first we had certain infectious agents and certain microbiome that, suffer a major change because, of the agricultural revolution that, had the possibility that these small groups stay in the same place and become larger groups and start to farm and of course would have, a different way to deal with let's say the feces of animals and the feces of the other people and the contact with different infectious agents. So we start to have an explosion of different infectious agents compared to the ones that we had before the agricultural revolution. And we start to have more elements, more orophacal transmission, and certain infectious agents that we are not used to, such as Rota, Corona, Paramyxio, and influenza virus, smallpox, and so on. And then it came the second transition, that is the Industrial Revolution. And of course, we had, major changes. But if we think, about what were really the sort of like the change, and one could associate a major change with the Second World War II. And it's also what is considered, the Anthropocene when we think, about the eras. And in this case, so humans made a major difference in, in every aspect. And then we start to having, huge cities, treated water. We don't have much contact with animals, and hygiene and antibiotic and processed food, pollution, and so on. And this leads to a new change of the infectious agents that we we deal in a daily basis. So we start to have less helminths, less mycobacteria, less hepatitis, and also changes in the microbiota. So as you can see, in 10,000 years, but also in 70 years, we had, these major changes in terms of the infectious agents that we are associated with. And these led to a contemporary epidemic of allergic, autoimmune, and inflammatory disease. As you can see here, there is this inverse correlation, the reduction of certain infections from the Second World War II, and the increase of immune disorders, such as autoimmune disease and Crohn's disease, type 1 diabetes, allergic disease such as asthma. And inflammatory bowel disease is among the inflammatory and immune conditions associated with this huge increase in the past few years. And IBD, inflammatory bowel disease, is a chronic inflammatory disease of the gastrointestinal tract, and has two main entities, the Crohn's disease and ulcerative colitis. And it has several signs and symptoms and long-term complications, as you can see in these two tables. And it is the paradigm for IBD pathogenesis that both environmental factors, the intestinal microbiota, will influence and trigger inflammation in individuals that are genetically susceptible. So as you can see here, and as you know, the intestinal mucosa, it's this incredible space where, interactions are taking place, and we are, amazed by how the immune system and the microbiome interact in this in this region of our body. But as you can see here, we have, the microbiome. We have among these bacteria with some pathobionts, and both the innate and the adaptive immune system will be essential both to resist and control the certain bacterial and the bacterial numbers and the fungi numbers and the virus and so on, and also to contribute to the toleration of the presence of these infectious agents. And of course, as you know, there is another dilemma here, that is the fact that we need to deal with food that is this huge amount of antigen that we eat every day. And this, of course, will cause a physiological inflammatory response that deals with these conditions. However, due to these issues that we just discussed, such as the use of antibiotics, the type of diet, the hygiene, and so on, we observe an increase in terms of dysbiosis, and in this case, an increase in the number of pathobionts, and this eventually, depending on genetic conditions, will cause pathological inflammation. And it is interesting to observe that sick individuals, meaning individuals that have eventually dysbiosis and are eventually sick, they can transmit these pathobionts to other hosts. And there are several genetic changes that are associated with disease, such as IL-10 and IL-10 receptor mutations, NOD2 and ATG, as you know, are associated with the appearance of or increased susceptibility to this disease, and as we discussed, environmental issues. So many years ago, it was shown, and I think it's an interesting observation, that IL-10 deficient mice develop chronic enterocolitis. And what they observed was that the mice that were kept in conventional animal facilities, they eventually, after few months, they develop a severe disease, when they were kept in SPF conditions, the disease were mild, and when they were kept in germ-free environment, they had no disease. So this clearly indicates that the presence of the microbiome triggers disease in IL-10 deficient mice. And depending on the conditions of the animal facility, this would change. So it's clear that in this case, low tolerance to the presence of the microbiome in the case of IL-10 deficient mice is associated with disease. And in this case, putting that graph, as you can see here, even with less bacteria, IL-10 deficient mice is much more sick than wild-type mice. And we have been studying this cytokine, that is macrophage migration inhibitory factor, is a pro-inflammatory mediator that control different inflammatory mediators, such as cytokines and lipid mediators and nitric oxide. The receptor of MIF is CD74 and CXCR4. It affects different aspects of immunity, such as TLR expression, apoptosis, and so on. So what we observe in several studies and other groups as well was that, for many infections, such as viral, bacterial, protozoa, and helminth infections, MIF deficient mice would be eventually less resistant, so they would have more of these infectious agents in their body, but they were less sick. So this indicates to us that MIF deficient mice seems to be associated eventually both with resistance as well as with tolerance. And in this study that we did many years ago, we observed in a model of toxoplasma, in oral toxoplasma infection, that MIF deficient mice survive while the wild type die. And when we look to the pathology, as you can see here in the second graph, the size of the intestine was less affected in the MIF deficient mice. The histological score was reduced in the MIF deficient mice, as you can see in these figures. But then, surprisingly, the amount of toxoplasma was much higher in the MIF deficient mice. So this indicates that despite the pathogen burden, the animals were less sick. So this indicates to us that these animals are more tolerant to infection. But this is not only associated to infection. As you can see in this colitis model in RAG deficient mice, that we transfer CD45RB CD4 cells. So these animals, when are RAG deficient mice, they develop a colitis, as you can see here in the black bars. And when the recipient animals were deficient of MIF, they had less disease than the ones that were sufficient for MIF in the RAG background. And also treatment with anti-MIF reduced colitis in a similar model and in a chemical-induced model induced by DSS. So this clearly showed that MIF seems to be part of the mechanism that triggers colitis in these different models, and the absence of MIF protected. So then we came with this very kind of naive question that would be, so what is dominant? High or low tolerance to inflammatory bowel disease? And another way to ask this question would be, is MIF involved on IBD development in the IL-10 background, deficient mice background? So the idea was very simple, just to cross the MIF deficient mice with the IL-10 deficient mice and see what we got. So basically that was the idea. And then my student came to me after 2 weeks and said, Marcelo, something really weird is happening. I put, two mice two females of IL-10 and one male of MIF, or vice versa, and to cross, and all the IL-10 animals die. And I said, oh, that's really curious. Why don't you do this as, a real experiment? go there, put your boxes of crossing, and weigh them, every day or every 2 days, and then let's see when they die, if you can reproduce that. And then, they came with this observation. As you can see here, in the IL-10 alone, they gain weight and they survive. The ones that were maintained with wild type had some difference in terms of weight loss, while the animals, the IL-10 deficient mice that were kept with MIF deficient mice, they lost weight and they all died, about 2 weeks. So we were surprised, and this is very robust and very consistent. And when we do the co-housing of MIF wild type, there is no weight loss or lethality, showing that the animals must have a genetic susceptibility to lose weight and to die when co-housed with MIF. And as you can see here, the animals that were co-housed with the IL-10 deficient mice co-housed with wild type, they were fine. So when we did colonoscopy, as you can see here, they are all IL-10, okay, or just IL-10 or IL-10 that were co-housed with MIF in the middle, or IL-10 that was co-housed with wild type. And as you can see, the ones that were co-housed with MIF had the rectal prolapse, and they also, when we look to the colon by colonoscopy, we observe bleeding and absence of feces. When we do the macroscopic analysis, there is a major reduction in the intestines length of IL-10 deficient mice co-housed with MIF, and a major pathological change, as you can see here, in a complete change in the structure of the intestine. So we look to neutrophils and eosinophils. We observe that the IL-10 deficient mice co-housed with MIF had an increase on neutrophils and eosinophils in the lamina propria. They also present a significant increase in interferon gamma positive cells, and some increase in double positive interferon and IL-17 positive cells, with a clear increase in Th1 cells in this case. We observed that the animals also present an increase in FOXP3 cells, CD4 FOXP3 cells, but of course these are IL-10 deficient mice, so these Treg cells are unable to produce IL-10, but still they are increasing in the animals that has more inflammation. And these cells, they do have more TNF. So we found that the Tregs that are present in the lamina propria of IL-10 deficient mice have an increase on TNF. So we asked whether the disease triggered by co-housing was dependent of infectious agents. So the hypothesis was this tolerant genotype would harbor more pathobionts in their microbiome. So we did fecal transfer, like just transferring the pellets or doing a gavage. And as you can see here, well, just the feces is sufficient to cause the disease. And when we filter the feces, blocking the passage of bacteria, we observed, and fungi, we observed that in this case, the filter feces was not able to trigger disease, suggesting that, a microorganism larger, bigger than a virus would be associated with this disease. So we analyze the microbial diversity with the idea that an animal that would be more tolerant maybe would have a different microbiome, suggesting that they would have more diversity. And that's exactly the case, while IL-10 has the less diversity and wild type is in between. as you can see here, both in the beta and the alpha diversity. So MIF deficient mice or tolerant mice have a bacterial microbiome composed of more different taxa. And what was interesting was that IL-10 deficient mice by itself, before developing any disease, they have a microbiome that is enriched by taxa that are associated with a health condition, while the MIF deficient mice had more bacteroidetes than Firmicutes. And in the case of IL-10 was the other way around. And then we asked whether this microbiome would interact and how would they interact with the immune system. So we analyzed the presence of IgA in the bacteria of these different groups of animals. And it's very clear and it is striking that so MIF deficient mice had the bacteria with much less cover by IgA, but much less than wild type, that was less than IL-10. So IL-10 deficient mice had a huge amount of their bacteria covered by high amounts of IgA compared both to wild type and to especially to MIF deficient mice. So this indicates that IL-10 deficient mice had more IgA covering their bacteria, while MIF is in the other spectrum. So we decide to analyze how the co-housing would change the IL-10 microbiome, and basically what we see is an increase in terms of diversity. So the IL-10 had less diversity. After the co-housing, they now have a microbiome that is more diverse than before. And we observe a change in terms of this microbiome after a co-housing with MIF. And it's interesting to observe that this change is mainly associated with a loss of bacteria that are considered markers of health. So this suggests to us that the bacterial composition would be responsible by the disease, and we decide to do a treatment with a cocktail of antibiotics, ampicillin, vancomycin, neomycin, and metronidazole. But as you can see here, this treatment, and we did in different ways. So we treat just IL-10, just MIF and IL-10. So everything we did was insufficient to protect the IL-10 deficient mice during the co-housing. So this was a problem. So our hypothesis was that bacteria was responsible, but in this case, we couldn't observe that. So we look and were able to identify different groups of bacteria after co-housing in these antibiotic-treated animals. And we isolate some of them, and we found that they are susceptible to gentamicin. So we are currently doing these experiments to see whether gentamicin can protect against the effect of co-housing. But then we go back to our very first experiment, that was the back crossing. So again, let's see what's happened when we have the double knockout. And, because we were having this problem of animals dying, so we ended up doing this type of back crossing, like a MIF deficient mice, IL-10 heterozygous, crossing them, and eventually we would have a double knockout. So let's see what's happened. And again, we had a major surprise. So we thought that the double deficient eventually would be better than the single deficient IL-10. But what we found was that, we had this expected ratio based in a Mendelian distribution of 25%. And what we found, expecting a bit more than 20 double knockouts, we only get 9. And of these 9, only 3 animals survived more than 4 weeks. As you can see here, the double knockout present a rectal prolapse, and they are much smaller compared to the MIF deficient and IL-10 heterozygous, the one that survive. So we asked whether the problem was associated again with the microbiota, because these littermates were littermates from animals that are basically MIF deficient mice, mothers and fathers. So we asked whether co-housing the double knockout with IL-10 would cause disease in the IL-10, and that's exactly what happens. And fecal transfer of these double knockout also cause disease in the IL-10 deficient mice. So at this point, our conclusion is that co-housing with MIF trigger colitis in IL-10 deficient mice, while type do not induce colitis in the IL-10 or become sick when co-housed with MIF. MIF deficient mice display a microbiome with increased diversity and presence of pathobionts, and we believe that this is the cause of triggering disease. We don't know exactly what species is responsible for that, or a group of bacteria or fungi. Antibiotic treatment was unable to prevent the disease, and we were able to identify some bacteria that survived the cocktail treatment. So at this point, we believe that low tolerance is dominant to disease development. So the fact that lacking MIF is unable to recover the disease indicates that low tolerance is dominant to the presence of pathobionts, because this double knockout was raised in an environment in which MIF deficient mice was present, so the parents. So then our final question at this point was if the disease triggered by high tolerance mice would be something exclusive to MIF deficiency. So we use different genetic deficient mice, and it's well known that certain deficiencies are associated with the presence of pathobionts and disease. But in all the cases I know, at least, the animals, the mutated animals themselves are also sick. And this is not the case for MIF, so they are really healthy. So we use the mutant mice, so the animals that do not have B cells, animals that are L18 deficient and TNF receptor deficient. And as you can see here, animals that are TNF receptor deficient, when co-housed with IL-10, also cause disease. So our hypothesis from this study is something that we are carrying out now. So is that, individuals that are more tolerant to intestinal tissue damage might harbor an infectious community with increased ability to cause disease in individuals that are less tolerant because, let's say, genetic reasons or certain environmental reasons. So more tolerant relatives of individuals with IBD might be the primary source of pathobionts associated with disease developed in susceptible hosts. So I would like to thank the people that did the work, some Vinicius Mendes and Letícia Martimiano. They are a PhD, and Letícia now it's also a PhD student. When she did this study, she was a master's student. Jamil, Helena, Andresa. This study had the essential collaboration with Heitor de Souza, Fábio Canto, Batiar, and Dan Littman and Varsha, and the financial support of CAPES, NPQ, FAPERJ, and Fulbright. So thank you very much. I will be happy to discuss with you these results.
So thank you again, Marcelo. As always, your talk was amazing. The results are impressive. It's always a nice story when we hear you talk, right? It's always very interesting. Yeah.
Yeah, I think it was intriguing, like from the beginning, from the very beginning to the end. we were like, oh my goodness. So from the crossing to this final, when we looked to what animals we got from the crossing, it was also quite surprising for us.
And I think it's always interesting, this discussion about disease tolerance and disease resistance. Especially, I'm working with TB now, for example, and most of the people live with the bacteria without developing disease.
Exactly.
Just a little bit of them develop an inflammatory disease to the pathogen, right? So this is very interesting. So we're going to switch to Portuguese now for the questions. Então as perguntas vão ser em português. Lembrando, os alunos têm prioridade aqui as perguntas, mas enquanto eu tenho algumas perguntas também que eu fico interessante. Eu sempre acho, sempre que a gente vê esses dados de teoria da higiene e aumento de aparecimento de doença autoimune, doença autoinflamatória, quando tem menos exposição aos patógenos, eu sempre fico em dúvida até que ponto, de fato, a gente tem... Quando que a gente consegue dividir esse aumento de doenças autoinflamatórias e autoimunes ser simplesmente um aumento da descrição ou caracterização da doença e aumento da notificação de casos, e etc., que ocorreu mais recentemente, versus, de fato, a gente ter uma mudança tão drástica na microbiota, na exposição à micobactéria ambiental, etc. É só uma discussão, não tem muito a ver com os resultados, isso por enquanto, mas é uma coisa que sempre me intrigou. Porque algumas coisas não batem, né? Tipo, a gente está tendo um aumento também de asma e etc. em países subdesenvolvidos que ainda têm uma condição mais precária, etc.
Mas acho, Diego, assim, existem determinados aspectos que claramente caracterizam o que a gente chama de antropoceno, né? Assim, se você pega uma cidade no Brasil, uma cidade na África, uma cidade... É claro que elas não são idênticas em termos de certas características de higiene com algumas cidades na Europa, nos Estados Unidos, mas ainda assim elas vão ter certas características de convívio que são muito diferentes de um convívio rural. Isso assim, é difícil a gente questionar isso, né? E quando a gente olha, sei lá, a indústria de alimentos também, né, a gente vê que esse fenômeno, né, que você menciona, por exemplo, né, vamos pegar a obesidade, por exemplo, ele também é um fenômeno que está distribuído, né, e por razões que a gente começa a identificar claramente. Então, a indústria de alimentos tem um papel central na obesidade mundial, nos países desenvolvidos, subdesenvolvidos, nas classes altas e, principalmente hoje, nas classes baixas. Então, assim, certamente, Diego, a identificação e o diagnóstico influenciam no número de casos que são reportados, mas, assim, as notificações nos últimos 40 anos, elas seguem mais ou menos o mesmo padrão de controle. E, claramente, o aumento ele não se deve a unicamente ou principalmente a diferença de notificação, indicando que realmente existe um componente epidêmico de doenças imunomediadas, né? Agora, se trata de uma questão de eumintos, de micobactérias, de vírus, provavelmente esses diferentes componentes vão ter um papel central nos diferentes ambientes, nas diferentes regiões. Então, eu estou bem convencido de que esse é um fenômeno robusto, sabe, Diego?
Obrigado. Tem aqui duas perguntas, da Vanessa e do Guilherme. Se a Vanessa não se importar, eu vou falar para o Guilherme fazer a pergunta primeiro, porque ele é aluno do programa, a gente está dando preferência para as perguntas dos alunos.
Claro.
E aí a Vanessa pode perguntar também, tudo bem? Se você quiser abrir o microfone, Guilherme, fica à vontade.
Tudo bem. Professor Marcelo, muito obrigado pela palestra, foi muito importante, muito boa. Não sei, só um pensamento. Será que seria importante, talvez, colocar nessa situação uma questão de tolerância, de imunidade treinada? será que essa microbiota também estaria influenciando na imunidade treinada? E isso influenciaria também nesse fenótipo?
Eu acho...
Porque,
Desculpa, pode terminar.
porque é uma coisa muito complexa, né? É a microbiota, é a tolerância, é a resistência. Não sei se na resistência entra a imunidade treinada. Será que existe também a influência da imunidade treinada nesse fenótipo? Será que de treinar a célula imune para tanto responder muito forte ou ela ficar tolerante ao LPS por conta da microbiota estar ali, ela ser mais tolerante, participar mais da tolerância ao dano e não tanto na resistência?
É super importante, Guilherme. Assim, a gente olhou o IgA nesse momento por razões mesmo técnicas. Tipo assim, nós estamos olhando os peptídeos antimicrobianos, nós estamos olhando, por exemplo, a IL-22. Então, isso que você fala, eu acho que é super relevante. A imunidade inata num ambiente que é de contato contínuo com a microbiota induz um estado que é diferente, obviamente, do estado da resposta imune inata em tecidos que, em princípio, são estéreis. E eu acho que essa é uma questão muito interessante e é pouco ainda, no meu conhecimento, é pouco explorada. Quer dizer, essa questão, existem dados interessantes, artigos mostrando, inclusive, esse aspecto de imunidade treinada em células epiteliais, que vai bem nessa linha que você está levantando. Mas eu acho que assim os estudos de imunidade treinada nesses ambientes com grande quantidade de micro-organismos ainda é uma coisa, me parece, pouco conhecida. Um outro aspecto que eu queria talvez realçar e que tem a ver com a sua pergunta também é a definição mesmo de tolerância, né? Assim, já me vem pelo menos 3 definições de tolerância que são conceitualmente diferentes e talvez o nome tolerância acabe confundindo, né? Uma é isso que a gente discutiu, disease tolerance, é a menos conhecida. A segunda é a tolerância imune, que é dependente da imunidade adaptativa, né? Por exemplo, o papel de células T regulatórias, né? Ou a morte de clones. E a outra é isso que você levanta, né? Que é a tolerância ao LPS, que é a não responsividade depois do estímulo com o LPS, pelo menos por uma janela de tempo. Então, assim, a minha sugestão para os alunos é talvez voltem lá e comparem o conceito e o significado de tolerância, porque eu acho que isso é super importante, mas está aí ainda num momento meio de confusão, porque esses conceitos são diferentes, mas recebem o mesmo nome. Mas muito boa a sua questão e a gente está olhando para isso, tá, Guilherme?
Obrigado, professor. Realmente a tolerância, hoje em dia, ela é bem dissecada, né? Existem esses 3 tipos mesmo.
Vanessa? Ou, não sei, Diego, desculpa aí
Tem uma outra pergunta de um aluno que, infelizmente, a gente está com o tempo bem apertado. Eu não sei se... Eu vou passar essa pergunta, se a Vanessa... O que você acha, Vanessa?
Eu abri aqui para te parabenizar e que sempre é muito bom te ouvir. A gente aprende muito, particularmente gosto bastante do tema. Vou deixar o Diego fazer a pergunta do aluno, que é a prioridade aqui da atividade. Se tiver tempo, depois a gente
conversa. Vou tentar responder rápido, assim. Vou tentar ser...
Eu acho que a gente só vai ter tempo para mais uma pergunta, infelizmente, porque vai começar a sessão oral agora, já daqui a uns minutos. Mas é a pergunta do Jefferson que ele mandou aqui para a gente. Ele está parabenizando pela apresentação e perguntando o seguinte: como você comentou, em muitos modelos de doenças autoimunes se observa a correlação com disbiose. Em humanos, você acredita que o desequilíbrio da microbiota é a causa ou a consequência dessas doenças? Porque há diversas teorias sobre esse assunto e eu gostaria de ouvir o seu ponto de vista sobre essa discussão.
Essa também é uma questão super importante, né, Jefferson? É claro que a gente tem que deixar claro o seguinte: os camundongos, eles são coprófagos, quer dizer, eles comem fezes. Isso, para começar, é uma diferença importante. Ou seja, esse convívio entre animais que comem fezes obviamente, vai ter um impacto muito significativo. Agora, entre humanos, a gente também sabe que nas famílias e você tem composições microbianas que são bastante correlacionadas, né? Então, como eu estava discutindo, diversos aspectos vão interferir, sim, com o microbioma. Então, a dieta, o contato com outros indivíduos, o uso indiscriminado de antibiótico, a ausência de contato com animais. Então, assim, existem diversos fatores ambientais que influenciam a microbiota. O que a gente está mostrando aqui é que, apesar de tolerância não ser exatamente baseado no controle dos micro-organismos, o que a gente observa é que, se você é tolerante ao dano, eventualmente você é capaz de ter uma microbiota que tem mais patobiontes. Então, eu acredito que os fatores ambientais e os fatores genéticos influenciam a microbiota, que, por sua vez, vão influenciar o desenvolvimento de diferentes doenças, sim, autoimunes, alérgicas e autoinflamatórias e inflamatórias.
Gente, infelizmente o nosso tempo está curto. Eu acho que eu teria mais perguntas a respeito de imunidade celular. A Vanessa também, a gente pode mandar aqui para discutir.
Vai ser um prazer depois responder.
Então, se tiver mais alunos que tiverem perguntas, quiserem mandar para o e-mail do workshop, a gente encaminha para o professor Marcelo. O professor Marcelo é ótimo, certeza que ele vai adorar interagir com os alunos. Queria agradecer mais uma vez, Marcelo. Foi ótimo, mais uma vez, frisar aqui. Obrigado por colaborar sempre com o nosso programa, sempre atende os nossos convites, participa aqui, acho que todo mundo adora.
Um privilégio, uma honra, Diego. Eu tenho muitos amigos queridíssimos aí, você é um beijo, Vanessa, Fernando Cunha, Zeca, Tiago, Dario, meu Deus, tantos, Larissa, eu adoro.
Infelizmente não pudemos fazer presencial, como você disse, né? Seria mais legal a gente interagir em pessoa, mas vamos tomara que tenha uma próxima oportunidade logo aí que a gente possa se encontrar, né?
Diego, super obrigado, gente. Um abração, boa atividade aí para vocês, bom encontro.
Obrigado, Marcelo. Forte abraço.
Abração.
Pessoal, então agora a gente vai migrar para as apresentações orais, certo? Então, obrigado a todos que participaram dessa sessão. Vamos começar agora com as apresentações orais. Até mais, pessoal.